Hello everyone, my name is Cindy Lopez and I will be presenting the Golgi apparatus. Here's an image of the Golgi apparatus taken from our textbook, Becker's World of the Cell. From this first image, we can see that a few features pop out. There are these flattened vesicles or stacks that are known as cisternae. They are formed by budding off from the endoplasmic reticulum. The buds at the bottom here of the image are the free vesicles known as transition vesicles, which go off to other components of the cell once they bud off from the Golgi apparatus. The Golgi apparatus is described as having three sections: the cis, trans, and medial Golgi. The cis Golgi faces the endoplasmic reticulum back here, or the top portion if you want to look at it from that way. The trans Golgi faces the plasma membrane at the bottom of this image here, and then the medial Golgi is in between the cis and trans Golgi. Then there's an image here to the right that kind of gives you more of an orientation of how the or where the Golgi apparatus is located at in comparison to the endoplasmic reticulum. The Golgi apparatus has an important role to play with modifying proteins. One of the modifications that I'd like to explain involves glycoproteins that are modified within the Golgi apparatus. They first begin their glycosylation, which is the process of adding a carbohydrate, inside of the endoplasmic reticulum. Once these glycoproteins have made their way into the Golgi, they can undergo through a process known as terminal glycosylation, which means that the carbohydrates formed earlier in the endoplasmic reticulum are removed. Once terminal glycosylation is completed, the protein is then free to move out of the Golgi apparatus and onto its next destination. It is important to note that this is not the only modification that occurs inside of the Golgi, as many other types of modifications do occur at the different cisternae in the Golgi apparatus. For the most part, what goes into the Golgi apparatus has to come out in some way, as we can see from the image here, demonstrating the journey of the lysosome. There are many diseases associated with the malfunction of the Golgi apparatus. Some include neurodegenerative diseases such as Alzheimer's and Parkinson's disease, infectious diseases like human rhinovirus and the ORF virus, cancers such as breast and prostate, and a few other diseases related to the pulmonary arterial hypertension and cardiovascular diseases. One of the diseases that I'd like to focus on is Parkinson's disease, which has been linked to a rise due to the fragmentation of the Golgi apparatus. There is a mutation in the leucine-rich repeat kinase 2, or LRRK2, that has been present in patients with autosomal dominant Parkinson's disease. LRRK2 is considered to be one of the most common genes linked to the disease. It has been shown that an inactivation of LRRK2 can lead to the fragmentation of the Golgi apparatus, and then it disrupts the vesicle trafficking in kidney proximal tubular epithelial cells. LRRK2 has been known to directly phosphorylate RABs, which is a GTPase that is involved in intracellular vascular transport. In this image, taken from the article, The Function of the Golgi Apparatus in LRRK2 Associated Parkinson's Disease, shows how LRRK2 can act as a center and attaches to RAB family proteins as well as others. It then affects the trans Golgi network, which is labeled as TNG, or the Golgi morphology. It can then generate the pathological features that are associated with Parkinson's disease.